It's a pleasure to be in this room. Normally it's for math talks. Today it's still for math talks, but for a different audience, so it's kind of a new experience. I'm going to talk about some work I've been doing, in fact, for over several years on relations between geometry of wounds and the way they heal. And it started by some in vivo work, essentially on Drosophila and zebrafish, which is mainly done with a team in Faculty of Medicine in Lisbon, the lab of Jacinto. And that then, as you will see, we have moved a bit recently also to working on cell monolayers, especially with the team of Benoît Ladoux here from Institut Jacques Monod and Mechanobiology Institute in Singapore. Because here we have better access to the physical parameters, so we can understand a little bit better what's going on in our systems and measure some things more directly. And for the math work, it's essentially done with a postdoc, Ibrahim, who is now Shedadi, who is in INRIA, in Montpellier now, and Patrizia Bagnerini from Genova. And then for the more theoretical stuff, it's done with Matteo Novaga, who is in Pisa, and Antonin Chambol, who is in Polytechnique. So what we're looking is at either zebrafish, either larvae or adults, or in Drosophila essentially at the embryo stage, so really at the very beginning of the development, and then at the pupal stage also, where the animal doesn't move too much since it's inside its enclosure, and so we can film it quite easily and get movies of how the wounds heal. And then for the monolayers, either MDCK, which is, these are canine kidney cell lines, or some human keratinocytes. So what's the phenomenon we study? We make a hole with a laser beam on an epithelia. So this is pupal epithelia in this case. And we see what happens here. What we're marking is myosin, so it's a protein which is affine to actin. And we're marking that with a fluorescent label, and we're seeing it showing up and then accumulating at the boundary. And what happens then is that you have this structure that forms at the boundary, so you have an accumulation of actin. Actin is a polymer, so it's F-actin, so filamentous actin that will accumulate all around your boundary. And that will be the main mechanism which is responsible for closing your hole when you are in an embryo. So what happens is what we should be seeing in this movie, if I can get it to run. You have that this actin cable, as we will call it, or purse string, will just contract its length. And by contracting its length, since the perimeter of the hole is going to zero, the hole will be closing. So this is the main mechanism for closing holes in embryos. And it has an advantage, and that's also maybe one of the reasons why zebrafish use it a lot, is that it's regenerative, so it leaves no scars at the end. So you really reconstitute, you do regenerative healing of your tissue, which is not the case in adult wound healing, as we know all. And we will be going back to that. So essentially, how do these actin cable work? So actin is also what you will have. Actin and myosin is what we're representing here as the red filaments are the actin, and the green structures are what we call the molecular motors of myosin 2. And that's also what works in your muscles. In your muscles, you have a very regular structure, or which is called a sarcomeric cable, where you have parallel actin filaments, these sarcomeres, and you have the motors just acting between the filaments. And so in a very efficient way, by moving along the filaments in both directions, they will, since you have a length of the motor, it will force this part and this part to move together. And so you have a contraction force generated. And this is a very efficient way of doing it. If you don't have a sarcomeric filament, but you have these sort of mixed polarity cables, then things are a little less clear that it works well, but still there has been a lot of recent work on that, and the things also work. So by moving along the filaments and pulling the filaments together, even if you have a less structured set of actin filaments, you make a contraction force this way. It's a less efficient one than the one you do with a sarcomeric structure, but still it's something which works, and it's this type of thing that you will have working on your purse string, so the actin cables we have seen in the wound healing films. And then you can also do it in higher dimensions, so you can do it for contracting surfaces by contracting 2D sets of actin fibers, or even higher dimensional sets of actin fibers, either in a sort of a homogeneous way or with some sort of anchoring sites at spot junctions, and then just contracting the junctions by pulling them towards each other, and that way contracting apical surfaces of cells. But essentially what we're interested in this talk is mainly this sort of mechanism. So you have the actin accumulating all around the boundary of your hole, and then by the action of the myosin motors, you're just contracting these filaments, and so you are contracting, making them move relative to each other. So contracting the perimeter of your hole, and by contracting the perimeter, closing the hole. So we have the first thing, and that was the first movie I was showing you on the left, is how these actin structures form. And this is a work that we published in JCB a couple of years ago. You can see that they form at a certain distance from the boundary. This is a time scale, so early times are in blue, late times are in red. You can see that away from the hole, there's sort of a random distribution of times. This is the time where the maximum goes through each pixel of the image. If you're away from the wound, it's sort of random, there's no information. If you're close to the wound, you can see that you have a maximum of actin at a certain time, which is what we call here the 0 time, which is the formation time. And then as time advances, you have this propagating to the boundary and finally giving rise to the actin cable that will be responsible for closing your wound. And at the same time, we realized that as the actin is propagating, you also have sort of a contraction wave. So if you look at this cell, when the actin goes through it, it will contract. And then when the actin passes to the next cell, so here's where you have the wound, it's propagating from left to right. When the actin goes to the next cell, then it will again redilate, and it's the next cell that will be contracting. So you have a mechanical wave, an actin wave, a myosin wave, giving rise to this cable, which will then be closing your wound. And in this talk, we will be mostly concentrating on how you close the wound and not on how you form the cable. So how do you close the wound? Well, the first thing we noticed, and this is a very old paper by Jacinto, I think still while he was a postdoc, is that people had realized that these cables sometimes did not form all around the wound, but only in part of the boundary. As you see in this picture, these are monolayers of gut cells, KCO-BBE, and you see that that essentially the filamentous structure, this cable structure, has formed on this part of the boundary, and then you have sort of a diffuse structure in this part here. So people already knew that somehow you can have different types of actin structures at the boundary, that you don't always have a cable going all around and contracting to close the thing. What people hadn't really realized, and that's where math comes in, is that essentially you put the filamentous structure of the cable in the parts where you have a negative curvature. So in the parts where, from the point of view of your tissue, you are concave, and in this part here, which is a part where you are convex from the point of view of your tissue, then you don't have interest, as we will see, in putting a cable, so you do something else. And that's what we will be describing in this talk. So it's essential, this relation between what is the mechanism you're using to close your wound and the boundary curvature, the local curvature of your boundary in wounds. And it's something, as we saw here, it's a monolayer, so it's an in vitro system. The films I was moving where it was showing was films from Drosophila, so in vivo systems. We also have films from zebrafish. So it's something that's conserved across species, across systems. And it's not only specific of wounds, because it's also something that you see in morphogenesis. This is zebrafish epibole, so where you have that essentially you have your primordial cell is sort of your inner sphere, that's underneath all of this. You have cells that will be differentiating, but not all over the sphere, but only at the animal pole, so sort of at the north pole of your sphere, and that then will have to migrate to come and cover the whole sphere, so the whole embryo. That's the way you're doing this outer layer of cells. It forms at the north pole, and then it migrates to envelop everything. And what people noticed is that they migrate essentially by increasing the number of cells while they're migrating along the northern hemisphere. But once they go through the equator, you have an actin structure. That's what we're marking here in fluorescence, that forms sort of an actin ring, which goes all around the sphere, sort of a parallel around the sphere. And by contracting, it's pulling your skin and helping it come to envelop the whole sphere. So you have the same type of phenomena of purse string contraction in things like dorsal closure in Drosophila. In lots of morphogenetic movements, you have this type of mechanism. And that's why it's also recalled in the wound healing in embryos. It's because all the machinery is there, and it works very well to form the structures, so it works very well to repair them also. Here, what's happening? You think, oh, but it's always a circle, so why do you saying this thing about curvature? It's because as a structure in R3, this parallel in the northern hemisphere or the southern hemisphere, it's always a circle, and it will have a curvature which will be the inverse of its radius. But if you look at it as something moving on the surface of the sphere, so you have a constraint, you can't move in R3, you're moving on the surface of the sphere. So it's a geodesic curvature that you should be looking at. And if you look at geodesic curvature, it changes sign once you go across the equator. Because if you try to contract the parallel here on the, while you are in the northern hemisphere and you take a geodesic will be essentially going through the inside of the, you take two neighboring boundary points, the geodesic between them will be going through the domain itself, while when you go beyond the equator, the geodesic will be going through the outside of the domain. So you have a change of convexity and a change of geodesic curvature when you go through the equator. And so there's something being played by curvature when you do in forming these cables, and we're trying to understand what it is. This is a wound done in pupa, so here in pupa with lasers we can do wounds which are from the beginning non-convex, and then we can check that essentially we're having the cable forming also on the negative portion, curvature portion of the boundary, and that on the positive portion, curvature portion, you have some other type of things that seem to be playing the essential roles. And all these things, this is macrophages, so we don't care about them. They're nice, but they're not part of our problem. So when we notice this thing about curvature and making or not making cable, or how to make surfaces involved, as mathematicians, and this is work we did already in 2012, we thought of, okay, so when you put a cable, essentially what you're doing is, if you have a constant tension along your cable, then the only things that, oh, I don't have a blackboard, that's a bad thing. The only thing that, if you consider a cable element, and you consider that you have the same tension pulling on both ends of the cable element, if it's straight, you have two vectors which are symmetric and which will sort of balance. But if it's curved, they will have the same magnitude, but they will be both pointing down, sort of, if the curvature is like this. So they will have sort of horizontal or tangential components that balance, but their normal components will be both pointing in the same way. And you will have, the more of that effect, the more the curvature is high. So you will have a force that is generated by a cable, which is naturally proportional to curvature, to local curvature of your cable. So we started looking at how do curves, if you are looking at subsets of R2, or more general objects, so n-1 dimensional objects in Rn, evolve by mean curvature. And that's what this theorem is about. So if you consider a set is essentially the position of your wound. It has a compact boundary which is sufficiently regular. This is your cable or your the boundary of your wound where the cable will be forming. Then you know that if you look at an equation where you make every point move with a speed which is the minimum between its local curvature and zero, so you will be saying, okay, where the curvature is negative, I move with the curvature. Where the curvature is positive, then the minimum will be zero, so I don't put any speed. This is sort of saying I'm putting the cable only where the curvature is negative and putting nothing where the curvature is positive, and seeing how this thing will evolve. And then we can prove that, in fact, this problem has a solution, and that you stay with in a set where you will have sets, compact sets, which will keep the same regularity over time when you flow, at least up to a certain time where you can develop a singularity, which in particular will be when you close the hole. There will be no more holes, so surely since you close holes in finite time, it cannot be something that will exist forever. And what you can show about these flows by just the negative part of the curvature is that if you take posterior time t2, then the position of the wound, or the set which is occupied by the wound, is strictly contained in the set that it had at the previous position. So you're closing a wound in a way that you never lose any territory. So you know that at any later time, you will always be inside the position where you were before. If you had put a cable all around, which means putting what we call an unconstrained mean curvature flow, this would no longer be true. And that is easy to see because if you look at, for instance, here, and you put a cable in this part here where you have a very big curvature, it will be pulling you inside with a high speed. Here where it's nearly flat, it will be sort of doing a little bit going in the inside, since you still have a slight negative curvature. But here where the curvature is positive, if you put a cable, then it would be pulling you outside. So a normal mean curvature flow here would have a natural tendency to round up everything, and so to open this part at the same time as it would be closing that part until everything becomes convex, and then everything will close. While if you just put the cable in the part with negative curvature, then you never go back. Even in the parts of the boundary where the curvature is positive, you will always be moving forward. And so that's what we're seeing here is in this, let's go through the theorem. We are saying that, in fact, for any time, if we start this type of negative mean curvature flow, and we, from the same set as a normal mean curvature flow, which is what we call with a tilde, then for every posterior time, we will have that the wound will be smaller by using this method than by putting a cable all around, which is not obvious, because in the initial times you could sort of be flowing back, but then since you are getting a negative curvature everywhere very fast with the mean curvature flow, you could sort of catch up and even move faster and close faster your hole. This theorem shows you that this doesn't happen. So this is the mathematical way of showing it. And then essentially, how do you interpret it? Yeah?
Just a quick question. Here, in the formal equation, can we...
Yeah. Yep.
So in the NMCF equation, I guess you might like to add a multiplicative constant.
Yeah.
Homogeneous in space.
Yeah, when you do math, you put everything equal to 1, but normally you should have the tension of your cable or your local tension. That's what should be multiplying your curvature term in the physical problem.
Exactly.
Then,
so if you have this kind of multiplicative constant, what condition do you require for the uniqueness and the existence and the
so this would be just what you would have from a constant flow, from a constant tension case. If you have a variable tension case, then if you don't have enough regularity of the way your tension changes, then you can create singularities even just by the tension
So
itself.
that's the question, what's the minimum regularity
normally we just work with the mean curvature without the constant. I think if you have a nice C infinity tension and you have some lower bound on it, it should be okay. But if not, you can, I think you can create counter examples quite easily.
So C infinity and let's say vanishing.
Then if you don't have, if you put, because essentially what you prove, even for this, you can't work directly with this equation. You have to actually prove these theorems, you have to work really in the set formalism type of, because the equation doesn't make sense if you try to, then you need a lot more regularity if you want the equation to make sense always. So you have to work in a lot more abstract setting, where for the specific case of curves, you get this, but it's very delicate. And in fact, it's an existence theorem. It's a unicity theorem, but where the existence part is really very, very weak. It's in very weak classes, so it's not that tough. So the thing that you have to retain from the biological point of view is that essentially the fact that you have a monotonicity, so the fact that you have that the set at the posterior time is always inside the set at the previous times, it's just saying that using this type of negative curvature flow is a natural way of closing without ever losing any previously conquered area, which is sort of an interesting thing from the biological point of view. And then the fact that it is a more efficient strategy compared to putting a cable all around the thing, all around your boundary, and that's just the comparison result with the mean curvature flow. So you get those two things from the math. Now, the thing is that this is considering you have a cable where you have a constant tension and which is isolated. So it doesn't have to drag a tissue behind it. That's what that theorem would be about, in case you say that the force is just the curvature. But in fact, in reality, you know that the cable is attached to the tissue. In fact, it forms inside the tissue. So you have to take into account the mechanics of the tissue that it has to drag. And so all of that will have to be considered and see what would change in that. The problem is that then the type of analytical result that we have in that theorem, we are no longer able to prove it. What we can do is that, in fact, we can go and try to see what would be the simplest mechanical model so that we get more or less realistic results, and that's what we'll be talking about in the rest of the talk. But still, we see that this idea of putting cable mainly in the positive, in the negative curvature regions is something which really happens in nature, where the cables are having to drag tissues. So there should be something of this sort of ideal toy model that should be surviving in the real system. Then the other second problem is that I've been just talking about this cable contraction, so this purse string mechanism, where by contracting the cable you get the edges to advance. But in fact there's another mechanism which is also going along, and this is what we were seeing in those, what we were seeing here, is that in this part of the boundary, what the cells are doing is, instead of making this cable tangent to the boundary, they're making what we call lamellipodia, which are actin structures, which are like claws that they form to go and grab into the extracellular matrix ahead of them, and then they pull themselves forward. So this is what we call lamellipodal crawling. It's you just send your claws in front of you, and then you pull yourself to advance. And this is a very robust mechanism. In fact, it's the mechanism that most of the time is used in adult cells, in adult wound healing, where it has one advantage, is that since you're always sending your claws ahead of you into the open space, you're always pulling yourself forward. So even if you are in this positive curvature section, if you use that mechanism, you will be moving in the right direction. While here, the cable pulls you in the right direction, that would also pull you in the right direction. In this part here, the lamellipodia pull you in the right direction. A cable, as I had told you, would pull you in the bad direction, so you would have interest in not putting it here. So if you consider that you also have this lamellipodal crawling, and that's what we're seeing here, and when we do sort of enlargements of what's going on here at this tip, we can see these lamellipodia. So this sort of, you have your cells here, and then ahead of the cells you have these actin structures that are going on to the extracellular matrix and grabbing to pull yourself ahead. And you can see it, so these are images from pupa, and then we will be seeing images from the monolayers. You will always be seeing that. So essentially these lamellipodia, you can also have a bit of them even in the parts where you have cable. So you can't exclude them completely, but essentially they are a lot more important in the parts where the cable is weaker, and then in the rest of the boundary where you have the cable pulling you forward. But somehow both mechanisms are sort of there. The only thing is that there will be parts where one is predominant, parts where the other one will be predominant. So the purse string will be giving you forces that are proportional to the curvature times the tension. So this is the constant that Amaury was mentioning, and which will be pulling you inside where the curvature is negative and outside where the curvature is positive. And the crawling, the way we model it, is also as a flow, but which is not proportional to curvature. So it's just a constant normal flow, which always pushes you towards the inside of the hole, so the outside of your tissue. And the difference between the two is that since this one is proportional to curvature, it tends to round up things, because things where curvature is very big move very fast, things where curvature is small move slowly, and things where curvature is negative would even move back. So everything tends to become round very fast. While the other mechanism, the crawling, since it just moves everything the same way towards the inside, it sort of has a tendency to keep the initial shape. So that's a geometric way of seeing the different actions of these two mechanisms, and that we will be taking advantage of to separate their effects. So essentially we're going to try to understand what's the role of the one and the other of these mechanisms, where and why each of them is predominant. We already saw with that theorem that essentially cables should be predominant where curvature is negative, and lamellipodia where the curvature is positive, but we're going to try to confirm all of that and understand it better. And so see a bit what's the crosstalk between the two mechanisms that regulate the action of one and the other. So essentially the model we proposed for taking into account the tissue that's behind and the lamellipodial crawling is summed up here. So we're just considering a viscous interaction, so a viscous model for our tissue. So cell-cell interaction being described essentially by the differences in their velocities relative to their neighbors. We are considering some substrate friction because we realized we need to include it for some of the problems. So that means when the epithelia is moving over either the extracellular matrix in vivo or whatever we put as coating in our in vitro experiments, there is some friction dissipating there. Then we have no problem with incompressibility, since we see that our cells are being able to change their surface. So it's essentially because you would have maybe a 3D incompressibility, but since things can spread out, then in 2D you no longer have that constraint, because you're just looking at the 2D information, since you're just looking at the cell edges from above, so their apical area. So we don't have this type of constraint. And then the special thing about the model is that you have these boundary forces acting on the cable, which one is the purse string force, which we put here in red, and the other one is the one that's generated by the lamellipodia. And so when we put all of this together, we just get this model, and this is what we really are simulating in the rest of this talk. So we just have a viscous model with a viscous parameter here, and where you just have the symmetrized velocity gradient as your viscous stress tensor. And then you have this sort of weird boundary condition where you're just saying that the boundary component, the boundary normal component, is given by your lamellipodial force density and your cable purse string force on the boundary. And then the momentum balance is just saying that you have these friction forces which are proportional to the local velocity acting on your system. And to motivate this and to actually be able to measure things a little bit more precisely than in vivo, we started working with Benoit Ladou's group in vitro systems. In these systems, what we do is that we have a PDMS stencil, so where we have pillars of desired shape. You put it on top of your wafer, and you let the cells grow around it, so they will occupy all the space except the space where you have the pillars of your stencil. And then at a certain point, you remove the stencil, so you leave the holes open, and you see how the holes close. And this is what we're seeing here. So these are different shapes of pillars, half circles, sort of moon-shaped, or this sort of M-shaped things. These are initial positions, and then we just see how these things evolve. And you can do periodic structures, so just with one film you can have lots of information of what's going on. You have the advantage also that you have direct access to the substrate, so you can play with the substrate properties, mechanical properties. You can play with the coating of the substrate, so the way the cells adhere to it. And then you can also put drugs in your media which can affect, for instance, blebistatin will affect your myosins, so the way the cable can contract. So you can inhibit contraction by adding a drug, or inhibit your cable, your lamellipodal components also, or eventually even play with the junctions between cells. And the other thing is that you can actually measure forces directly, and that's something that I will be showing in the follow-up. And what's the similarity between this system and the in vivo system that we were studying in the beginning is that if we mark actin, we can see that the cable is there. If we mark myosin, we can see that the myosin is there contracting the cable. So the mechanisms are essentially the same. So we can get from this in vitro system information about what's going on in our in vivo system. And then we can really play in a very precise way with the shapes of the pillars, since this is just PDMS, so you can make them in whatever shape you want. And so when you do that, you can do shapes where, so this sort of M-shaped thing, which is a non-convex shape, where you have a positive curvature region here at the tip of the M. Oh, this one is the other way around. And you have the negative curvature regions, which will be created by these things when they round up. You can play by putting even in the same hole part with negative curvature and a part with positive curvature, which are more or less constant in each of their segments, and so actually measure it. So this is the pillar, the mask or the pillar in the PDMS structure that you use to exclude the cells. Okay. And for instance, by playing with the coating, here it's the fibronectin. You can see that the speed at which the cells move will change with your fibronectin concentration. So by playing with the amount of fibronectin you put on your coating, you can have cells moving faster or less fast. And so in a certain way, as we will see, you can favor lamellipodia or favor your cable. At the same time, you can actually even look at how your cells are putting their focal adhesions. So by marking your adhesion proteins, So here you're marking in red your actin, so you're seeing your cable in red here very well. But then you're marking your paxillin with a green fluorescent protein. And so you can, by looking in detail, you can even see how your adhesions are oriented. And you can see that it's not the same way in negative and positive curvature regions. So in negative curvature regions, this is your edge, so this is tangential to the edge, and you have your adhesions more oriented at small angles relative to the edge, while in positive curvature regions you will have a lot of adhesions which will be oriented normal to the edge, which is what you expect from lamellipodia, since in a cable contracting, your forces are exerted essentially tangent to your edge. So you expect the stresses to be applied on cells tangent to the edge, and that's what you see in the negative curvature regions as your main behavior. In positive curvature regions, you have these claws pulling you ahead, so your stress is exerted normally to the edge. And that's what you're seeing in the positive curvature regions as being the main thing observed. So it's motivating the type of approach we're putting into our model. Then these are the pillars that Vincent was talking about, is that if you want to measure forces, one of the ways is to, instead of growing it on a continuous substrate, you have a substrate which is full of little pillars, which is what we're seeing here in white in this image. You put the coating just at the top of the pillars so that the cells don't go and attach on the sides of the pillars. And you're marking these, the top of the pillars with your fluorescent protein. And then the cells are marked in red here, and the cells are marked in green. And then when you do the movie and you see the cells moving on this, you can follow the way the pillars are moving. And so you're seeing how the cells are pulling on the pillars. And that, since you know the mechanics of the pillars, since that's just physical material, you can do the inverse problem and deduce what's the force that the cell is applying on the pillar. And by doing this, you can measure the forces that are exerted by the cells a long time on the pillars. And again, you see that they are essentially tangential or mainly tangential in negative curvature areas, and mainly normal in positive curvature ones. So again, this confirms what we are putting into the model. You can even do this in a continuous way with what they call traction force microscopy. Here it's a silicon wafer, so a deformable material, but a continuous one. And then you put markers inside your continuous material, and you let the cells pull on it. Since it's deformable, it will be moving when the cells, to respond to the forces the cells are exerting. And since you have the markers with PIV, so you can follow the way those markers are moving and get information, sort of a continuous version of what we were seeing in, with the pillars. So you get all these deformation fields of your substrate, which are giving you information about how your cells are pulling on the things. And again, you can see that, that the forces are stronger near the boundary, and that they are more normal in the positive curvature areas and more tangential in the negative curvature ones. So everything is sort of adding up to this type of picture of having curvature and mechanisms being related to each other. Again, here it's the fibronectin experiments. So you can change the amount of fibronectin. So this is what allows your cells to grab to the extracellular matrix. To put a cable, cells just need to grab to each other. They don't need to grab to the extracellular matrix. So the cable shouldn't be affected by the amount of fibronectin, or at least not significantly. But the lamellipodia should, because if you want to crawl on something, you need to really grab onto the substrate. If you can't grab onto the substrate, you can't pull yourself ahead. And so you would expect lamellipodia to be a lot more efficient when you have a lot of fibronectin, so a lot of matrix where to hold on, than when you have little fibronectin. And 20 million micrograms is our control situation. And so we see essentially in these movies is that if you put very little fibronectin, things round up very fast and close a little bit slower. If you put an intermediate amount of fibronectin, then things sort of evolve in a balanced way where they round up, but where the lower part here still was advancing a little bit. And if you have nearly no, if you have too much fibronectin, then the lamellipodia really take over and things don't even round up. It's essentially this normal flow that's your main mechanism. So, so again, we are in the spirit of the model. We can even do a little bit better than that. This is another paper that we published this year with Ladou's group. Here we actually not only put no fibronectin in this sort of star-shaped patch, we even put something else where they really can't, this pluronics, so that it's really non-adhesive. So cells really can't adhere there. Lamellipodia are useless. And what cells have to do in this case is that they really have to build bridges. So here, the place which is adhesive, we put it in, we represent it in red. Once you get onto the non-adhesive patch, you really have to float somehow. You have to bridge across that non-adhesive patch. And so the only mechanism that works there is your cable. And in fact, you can see that things round up and that close in a very, very round form. So again, we can confirm the type of separation between these two mechanisms. Still one more experiment, I think it's the last one, to confirm these things. If you put this plebistatin and you inhibit the myosin, then you should be perturbing the cable, since the contraction of the cable is done by these motors. And what you see when you do that is that, in fact, cable is inefficient, and that's the lamellipodia which have to actually close your hole. And in this case, where you're perturbing the lamellipodia with this R2-free component, then you see that things round up a lot, even if you are at the normal fibronectin. So really the two mechanisms are, they're not mutually exclusive, but they're being used in different ways depending on what are the conditions. This is still one experiment where what we did is we cut the cable at both sides, and this tip, which in normal situation moves very little since it has a balance between the cable that's pulling it back and the lamellipodia that's pulling it forward. When you cut the cable, you sort of free this tip, and you can see it advancing just like you see these leading cell type of mechanisms in Silberzan's experiments. So really these two mechanisms seem to be all the time there and being used one or the other depending on what is more advantageous in each situation. And here is the simulations of our models. So essentially we make the model run from the initial contours, and we can see that it sort of reproduces in a nice way, even visually. And then what we do to actually do the comparisons, we do curves of speed of the boundary, normal speed of the boundary as a function of boundary curvature. And what we see is that our simulation points, which are the black curve and the little dots, are in fact quite close to what we get from the experimental points. If we consider all the complications of the experiments, it's really a very good agreement that we're getting here. And in fact, the thing of having this model is a way of actually being able to separate in a very clear fashion the action of the two mechanisms, because in the model you just have to put the parameter for the cable, so the tension, to 0. You will see what just crawling would do with the model, or put the parameter of the lamellipodia to 0 and see what the purse string would do with your And so here you can actually see, and that's one of the nice things about the model, that if you had just crawling, then you would have this type of evolution. If you have just purse string, as you would expect, you just round up and close in a round way. And if you have both of them, you have sort of what you see in the experiments, which is a balance of the two. And so you can see what's only crawling is this blue line, which would make you something that does not depend a lot on curvature, just increases very slightly with curvature. Only per string is something which is very good when you have very negative curvatures, but which starts being not so good when you get close to zero curvatures, and which is actually bad since it pulls you in the bad direction when you have positive curvatures. And that in fact that the overall behavior, it's nearly very close to the arithmetic sum of the two effects, which is sort of a simplification that we were not expecting to see so clearly. So essentially what the study is giving us is a nice way of separating the two effects and of understanding what's the advantage of using one or the other mechanism in different types of situations, which is very coherent with what we got from the experimental results, and which is essentially saying that this per string, this actin bundle contraction, is playing the major role where you have low adhesion. So this is the low fibronectin type of situations, and where you have very negative curvatures, which is the case where you have small holes, because there, since it's 1 over the radius, your curvature, that will be where you will have very high curvatures. While the crawling should be playing a big effect, and that's what we see, when you have positively curved regions, when you have a lot of possibilities of adhering in a very efficient way to your substrate, and so when you have large gaps, which would mean very small curvatures or zero curvatures, then you would expect your lamellipodia, as you see, to play the major role. So you get this sort of general picture from this study, where you should be, for gaps in plasma membranes, which are very small holes, expecting to see some sort of actin cables forming, and that's what people saw in when they make holes in single cells even, so just in the membrane. When you go to gaps in tissues, then when they are small, you should have mainly per string. When they are large, you should have that the crawling should be playing an important role. And that when they start being very large, then it's essentially your crawling that will be doing the closing. And when you have expanding tissues, then this should be the crawling that's doing the work, and that's what people see. When you have single cells, then it's typically the case where your boundary has a positive curvature, then it's crawling that's useful. You have no interest in putting any cable in those cases. Well, future directions, there's one big difference between the two cases we considered, is that when we do the holes with a laser, we are wounding, so we are destroying cells in the tissue, so there should be signaling associated to that. When we're making gaps with these pillars, we're not destroying cells, we're just preventing cells from occupying that area, and then we remove the pillar and they can occupy the open space. So this can be a way of seeing the difference, of seeing the effects of those signaling, the stress signaling associated to the wounding itself. But this is something we haven't developed for the moment, and that we should be doing now. And that would be an easy way of doing it, is doing wounds in the monolayers with the lasers, because there you can compare that with doing the gaps in the same system and see what happens. So the feedback between the mechanisms is also something which is not that clear. So how do they cross-talk between the two of them to regulate and optimize the way of closing? And in particular, now we have a project of studying the role of your intercellular junctions on this crosstalk, because in vivo we have some data that is showing that they are playing a role. The tight junctions are playing a role in this crosstalk. And then, well, mathematically, we would always have the dream of controlling this system so that you can close holes and wounds in optimal ways.